Bismillahirrahmanirrahim. Students, in this lesson I will discuss about Mendel's laws of inheritance. Gregor Mendel was a monk and he was a priest in Austria. He developed the fundamental principles of genetics and he proposed that there are special factors in organisms which control the expression of traits and their transmission to next generations. These factors were eventually termed as genes. He selected Pisum sativum, that was pea plant, to carry out a large number of experiments. In his writings, he gave reasons for this selection and he used 28,000 pea plants for his experiments. Here are the reasons for the pea selection. There should be a number of different traits that can be studied. The organism should have contrasting traits. For example, for the trait of height, there should be only two different phenotypes, that is tallness and dwarfness. The organism, if it is plant, should be self-fertilizing, but cross-fertilization should be possible. The organism should have a short but fast life cycle. All these features are present in pea plant. Normally, the flowers of pea plant allow self-pollination. Cross-pollination can also be done by transferring the pollen grains from the flower of one plant to the flower of another plant. Each trait study in pea plant had two distinct forms. Mendel succeeded in his work not only because he selected the right organisms for his experiments, but also because he analyzed the results by using the principles of statistics. True breeding. What is true breeding? The term true breeding means homozygous, and this term is used to describe organisms that produce offspring identical to themselves if allowed to self-pollinate. Monohybrid cross. Mendel studied the inheritance of seed shape first. For this purpose, he crossed two plants having one contrasting trait, that was seed shape, and monohybrid cross is a cross in which only one trait is studied at a time, and he crossed the true breeding round seeded plant with the true breeding wrinkle seeded plant. All resulting seeds of the next generation were round, and he declared the trait round seeds are dominant while the wrinkled seeds are recessive. The following year, Mendel planted these seeds and allowed the new plants to self-fertilize. As a result, he got 7,324 seeds, out of which 5,474 were round and 1,850 were wrinkled. So the ratio was 3 round, ratio 1 wrinkled. The parental generation is denoted as P1 generation. The offspring of P1 generation are F1 generation, that was first filial, and the cross in F1 generation produces F2 generation, that was second filial. Similarly, when true breeding tall plants were crossed with true breeding short plants, all offspring of F1 were tall plants. That is, tallness was a dominant trait. When members of F1 generation were self-fertilized, Mendel got the ratio of tall to short plants in F2 as 3:1. Mendel concluded that traits under study were controlled by discrete factors of genes. In each organism, the genes are present in pairs. During gamete formation, the genes or allele of each pair segregate from each other, and each gamete receives the genes from the pair. When the gametes of male and female parents unite, the resulting offspring against gets the genes in pairs. These conclusions were called the law of segregation. The next is Mendel's law of independent assortment. In the next crosses, Mendel studied two contrasting traits at a time. Such crosses are called dihybrid crosses. He performed on two seed traits, shape and color. Di means two. The trait of round seed seeds controlled by allele capital R was dominant over wrinkled controlled by allele small r seeds. Similarly, yellow seed color controlled by capital Y was dominant over green controlled by small y. Mendel crossed a true breeding plant that had round yellow seeds capital R, capital R, capital Y with a true breeding plant having wrinkled green seeds small r, small y, small y. All seeds in F1 generation were round yellow. When F1 seed grew into plants, they were self-fertilized. This cross produced seeds with 4 phenotypes. There were 315 round yellow seeds, 108 round green seeds, 101 wrinkled yellow seeds, and 32 wrinkled green seeds. The ratio of these phenotypes were 9:3:3:1. So here is a Punnett square, which is a diagram that is used to predict an outcome of a particular cross or breeding experiment. It is named after R.C. Punnett, an English mathematician. The gametes of both parents having all possible genetic setups are determined. A checkerboard is used to cross all the possible gametes of one parent with all the gametes of other parent. In this way, a biologist can find all the possible genotypes of offsprings. Mendel explained that two traits, seed shape and seed colors, are not tied with each other. The segregation of capital R and small r alleles happens independently of the segregation of capital Y and small y alleles. From his second experiment, Mendel concluded that different traits are inherited independently of one another. The principle is known as the law of independent assortment. It states as the alleles of a gene pair segregate or get separated and distributed to gametes independently from the alleles of other gene pairs. The parental generation is denoted as P1 generation. The offspring of P1 generation are F1 generation, that was first filial, and the cross in F1 generation produces F2 generation, that was second filial. An example of codominance is the expression of human blood group AB. The ABO blood group system is controlled by the gene I. This gene has 3 alleles IA, IB and small I. The allele IA produces antigen A in blood and the phenotype is blood group A. The allele IB produces antigen B in blood and the phenotype is blood group B. The allele small I does not produce any antigen and the phenotype is blood group O. The allele IA and IB are dominant over small I. When there is a heterozygous genotype of IA-IB, each of the two alleles produces the respective antigen and neither of them dominates over the other. So here is a table in which genotype, antigen produced, phenotype and relationship between alleles has been shown. Now I will discuss about incomplete dominance, which is a situation where in heterozygous genotypes both the alleles express as the blend, that is mixture, and neither allele is dominant over the other. As a result of this blending, an intermediate phenotype is expressed. Following is the familiar example of incomplete dominance. In 4 o'clock plants, the 3 flower colors are red, pink and white. There is no specific gene responsible for producing pink flowers. In 4 o'clock plant, the trait of flower color is controlled by 2 alleles. Let us call them capital R and small r. The true breeding plants capital R and small r have red and white flowers respectively. When a homozygous red flowered plant capital R is crossed with a homozygous white flowered plant, the heterozygous capital R small r plants of F1 generation produces pink flowers. Pink is a blend of red and white colors. This result clearly indicates that neither of the flowers allele capital R and white flower allele small r is dominant. However, when two heterozygous plants with pink flowers capital R small r are crossed, F2 generation shows phenotypes of red, pink and white flowers in the ratio 1:2:1. So here was the parental generation. Then F1 generation, in which plants with pink flowers, then they are crossed. Then there was F2 generation, that was a combination of red, pink and white in the ratio 1:2:1. So that was all about Mendel's laws of inheritance. I hope this lecture was a good source of information. Thank you and have a nice day.